Basic medical sciences, welcome. In the previous videos, we covered the gross anatomy of the pancreas and the histology of the pancreas. In both of those videos, we mentioned that the pancreas has two parts functionally, the endocrine and exocrine. So you can click the link on the top right corner and watch our playlist on the pancreas. In this video, we're going to focus on the exocrine pancreas. Right, so the pancreas secrete a pancreatic fluid or pancreatic juice, which is isotonic. And in this pancreatic juice, you find three important components: digestive pancreatic enzymes, electrolytes, and water. Pancreatic proteases are important for digestion of proteins, and they are secreted as inactive forms called zymogens or proenzymes, and they are secreted by the pancreatic acinar cells into the pancreatic duct. And these pancreatic proteases include trypsin, chymotrypsin, elastase, and carboxypeptidase, right? So the proenzymes for trypsin and chymotrypsin are trypsinogen and chymotrypsinogen respectively, and they are activated in the duodenum, right? And I said a trypsinogen is activated into a trypsin by what? By an enzyme, a brush border enzyme called enterokinase, right? So the enterokinase is on the mucosa of duodenum and jejunum, who will convert trypsinogen into trypsin. Then trypsin will convert other enzymes, like it will activate chymotrypsinogen to form chymotrypsin, and furthermore convert additional trypsinogen molecules into trypsin. So this is known as a positive feedback loop. Elastase, it digests elastin fibers and again is activated by trypsin. Carboxypeptidase, also activated by trypsin. Right, so the next group is, this one is very important, you need to remember it, the pancreatic amylase. Pancreatic amylase is for digestion of carbohydrates, and this one is secreted as an active form, right? The proteases we discussed are produced as inactive. Why? To prevent autodigestion of the pancreas, which may lead to pancreatitis. The next one is pancreatic lipase for digestion of lipids. The next one is phospholipase A for digestion of phospholipids, and nucleases for digestion of RNA and DNA. Number two, I mentioned the electrolytes. So the most important ones are sodium, potassium, chloride, and bicarbonate, right? And we'll talk more about this later. The concentration of chloride and bicarbonate increases with the rate of pancreatic juice secretion. And the third component is water. Right, one clinical significance, cystic fibrosis, right? So this is a disease caused by a defective chloride channel that impairs pancreatic water and electrolyte secretion. This will result in obstruction of the pancreatic ducts with the subsequent maldigestion and malabsorption. Let's talk about the feedback mechanisms, that's regulation of the production of this pancreatic juice, right? So this pancreatic juice production is stimulated by two main enzymes, the cholecystokinin, CCK, and secretin, right? So the cholecystokinin is produced by the enteroendocrine cells of the duodenum, the eye cells, and secretin is produced again by the enteroendocrine cells from the duodenal cells. This will be S cells. So you can see the pathway of cholecystokinin here. It has two pathways actually. You can see firstly for release of these enzymes and proenzymes, and this one it will stimulate the activity of secretin on the production of water and bicarbonate, right? So what's the function of secretin? This production of water and bicarbonates, and this pathway is stimulated by the cholecystokinin, right? So that's the stimulation. What about inhibition? Inhibition starting. So this process is inhibited by somatostatin. Thank you so much. If you like this video, please don't forget to give it a thumbs up and leave a comment in the comment section. And most importantly, subscribe. Peace out.